the Immune System, Innate Immunity by Laureano Andrade Vicente and the MCAT Disciples. So I'm very happy to give this lecture. It's one of my favorite topics. Okay, this is the AMC topic list. Okay, we're going to break this up into two lectures, Innate Immunity and Adaptive. And so today we'll begin with a general overview of immunity, and then we'll get into innate immunity. And then next lecture we'll do adaptive. Immunology is the study of how the body differentiates between self, non-self, and damaged cells, and subsequently how it rids itself of non-self or damaged cells. Immunology is a subset of physiology. It's very complex. It can be very, very complex. There's so many different pathways and so many different crisscrosses that occur in immunology, but I'm going to try to make it as simple as possible. Alright, so the anatomy of the immune system. You can break up the immune system in terms of anatomy into lymphoid organs and cells. Okay, and you can further divide lymphoid organs into primary and secondary lymphoid organs. So the primary lymphoid organs are going to be the site of immune cell birth and maturation, and the secondary lymphoid organs are going to be the site of antigen recognition and differentiation. Okay, in terms of primary lymphoid organs, you're talking about bone marrow, okay, and you're talking about thymus. The bone marrow is the site of B cell birth and maturation, and also T cell birth. Every single active immune cell is born within bone marrow. Every single innate immune cell is born in bone marrow. Every single red blood cell is born in bone marrow. Everything's born in bone marrow. Okay, every single cell that's found in blood is born in bone marrow. Okay. The thymus is the site of T cell maturation, okay? And you remember that TT. And again, the secondary lymphoid organs can be encapsulated or unencapsulated. That's just a terminology. Encapsulated would just mean that it's literally encapsulated, and it'd have a certain pressure associated with the inside of the capsule. Encapsulated would be like lymph nodes and spleen. Unencapsulated would be like Peyer's patches, gut-associated lymphoid tissue. So that's just lymphoid tissue surrounding the gut. Okay, Peyer's patches is a specialized sort of lymphoid tissue that has interesting roles in dendritic cells, extending out some extensions into the stomach. But let's not go there. Then there's a cell. Okay, you have innate immune cells and you have adaptive immune cells. So the adaptive immune cells are called lymphocytes. Both these two things together, both innate immune cells and adaptive immune cells, are the leukocytes, also known as white blood cells. You should know that distinction between a lymphocyte and a leukocyte. So every lymphocyte is a leukocyte, but not every leukocyte is a lymphocyte. Okay, so leukocyte is the general term for white blood cells, and lymphocyte is a specific type of white blood cell called the adaptive immune cells. Okay, so the B cells, T cells, okay, natural killer cells. White blood cells are leukocytes, including the adaptive immune cells, the lymphocytes. Okay, so that's the general classification. This is very specific to adaptive immune cells. Okay, so here's just a picture of the anatomy of the lymphatic system. I won't go through it. It's not important for us to know, but what is important for us to know is that the birthplace of the leukocytes, again, is bone marrow, okay, and it's also the site, bone marrow is also the site of B cell and NK cell maturation. Recall, bone marrow is a primary lymphoid organ. It is a site of B cell birth and maturation, okay. The thymus, again, is the site of T cell maturation, remember TT, okay. And we also have in this figure lymph nodes. Now, lymph nodes function to filter lymph. And what a great thing that is, because recall, the function of the lymphatic system in part is to pick up extra filtrate from the capillary beds, right? And that extra filtrate is going to have whatever was in blood. And whatever was in blood may be able to stimulate an immune response. And so what a great place to have a large collection of immune cells. Right? Because remember, this is a secondary lymphoid organ, the lymph node, and that secondary lymphoid organ's function is to have a large density of immune cells to recognize antigen and recognize pathogen to then exact an immune response. This is the structure of a lymph node. I don't care too much about it. I would just like you to know that the T cell zone is within the middle, okay, of this lymph node, and the B cell zone is within this follicle here. And there are different immune cells that co-localize with the different T and B cells to then exact a specific immune response. So again, the lymph node filters lymph collected from capillary beds and tissue, then the site, and is the site of antigen recognition. All right, we localize a bunch of different immune cells here so that we can recognize antigen, and then exact an immune response and differentiate from naive B and T cells into then effector B and T cells. Now recall, I don't think I mentioned this yet, but a naive B or T cell is a B or T cell that has not recognized the antigen yet, so they're naive. They are still fully matured, however, so the maturation has already occurred. Once they're in lymph node, and when they're in lymph node, then they're called naive B and T cells until they recognize an antigen that they recognize. An antigen is just a foreign substance that will initiate an immune response. Okay, that's what an antigen is. Alright, lymph nodes can be used as an indicator of the spread of infection upon physical examination. Again, I already mentioned this. Your physician, if you go to your primary care physician and you say you're sick, she'll often palpate some areas of your body, like under your chin, okay, under your armpits, to see if the infection has spread to that area or whether or not you have an infection, because they will often feel tender to the touch, or they may be enlarged or inflamed. And if they feel inflamed, that's because you either have a cancer within the lymphatic cells or you have something stuck within the lymph node. So you have an overproliferation of some cells, maybe. You have a lot of pathogens within the lymph node. Okay, again, it's a filter. A filter can get clogged, and when it gets clogged or when it gets inflamed, it'll feel tender and it'll feel enlarged. All right, again, function of the lymphoid organ, the spleen. The spleen is an example of a secondary lymphoid organ, and it is an encapsulated lymphoid organ, and it functions to remove aging and damaged blood cells and particles, such as immune complexes and opsonized microbes, from the circulation, and to initiate adaptive immune responses to bloodborne antigens. I'm not interested in showing you an image of the spleen. Just know that it functions to remove aging and damaged blood cells, red blood cells, and particles. Okay. Again, blood cells, red blood cells plus white blood cells, are derived from hematopoietic stem cells within bone marrow. And I'll go through some of the terminology, but I don't really have an image for you. Within the hematopoietic stem cells, when they divide, they'll divide either into a myeloid progenitor cell or a lymphoid progenitor cell. The lymphoid progenitor cells go on to make up the adaptive immune cells, so that B cells, T cells, and also the NK cells. Okay, the NK cells are related to the T cells, but are not exactly the same, but come from the same lymphoid progenitor cell. And the myeloid progenitor cell gives rise to the red blood cells, and it gives rise to any of the other leukocytes that are not part of the adaptive immune system. So, which we'll talk about soon, the monocytes, the neutrophils, the basophils, etc., etc., etc. So there it is there. That's what I just said. Again, this whole process is called hematopoiesis. It's just the birth and development of blood cells. That's what hematopoiesis is all about. In extreme circumstances, as I mentioned in the last lecture, it can occur in kidney, but primarily bone marrow. Okay, so here are immune cells in a nutshell. I'm going to go through them fast and loose here, and we probably won't have much to say about most of these ever again, but some of them we will. Again, we can't cover everything. It's immunology is a very complicated subject, and it can be much more difficult than what I'm about to make it, but I'm going to try to make it as simple as possible. The dendritic cell is a prophagocyte. So a prophagocyte is so when we say prophagocyte, a phagocyte is something that phagocytoses, bacteria or pathogens. So a phagocytosis is just when the cell wraps around the bacteria and swallows it. That is phagocytosis. When we say something is a prophagocyte, its main job is phagocytosis. So the dendritic cells found ubiquitously in tissue and plasma. It's related to the microglial cells in the central nervous system. If you've ever read about that, those microglial cells within the central nervous system serve a scavenger-like function, and it's one of the only scavenger-like functions that the brain has. Yeah, the red blood cells are obviously O2 carriers, secrete 2,3-BPG. We talked about that in biochemistry. When you have low O2 in the tissue beds, red blood cells start to secrete 2,3-BPG by doing a bypass of the glycolysis phase and give up the opportunity to make ATP to form a 2,3-BPG. Anyway, the megakaryocytes, they secrete platelets. Those that are born in bone marrow, just like the rest of these blood cells, and they secrete platelets. They're just these big, humongous cells that secrete platelets. You break off a piece of the megakaryocyte, and then you got platelets. And platelets function in coagulation, but they are not cells. Platelets are not cells. I know I've included them in the cells, immune cells, but they're not cells, but I include them here. But they are not cells. Prophagocyte is another prophagocyte, the monocyte, and it's found in plasma and differentiates into a macrophage when it's recruited to the site of infection. Okay, so that's the macrophage here, is what it differentiates into. The macrophage is the prophagocyte and resides in tissues and the extracellular fluid space, and will phagocytose bacteria and pathogens when they enter the extracellular fluid space. Okay, they also function to alert and recruit other leukocytes, particularly the neutrophils and the monocytes, to sites of infection by releasing cytokines that activate endothelium that, as we'll see, can then help to recruit neutrophils and monocytes. Alright, so the neutrophil. So the neutrophil is a prophagocyte. Okay, it's very important in phagocytosis, and it's recruited to the site of infection. Okay, that along with the monocytes are recruited to sites of infection, and then they clean up the debris and clean up the infection. All right, they phagocytose any bacterium that got into the cell. Another thing to note about the neutrophil is it's also called a PMN, a polymorphonuclear site. Okay, and that's just coming from the way in which it looks in the histological section. Its nucleus is polymorpher, if you would, and so that's why it's called that, a PMN. So we'll hear a lot more about him when we go to inflammation. You have the eosinophil, which is very important in dealing with parasites. That's about the only thing I'm ever going to have to say about it. What it does is secretes these toxic proteins and chemicals that will then deal with intracellular parasites. But we will never talk about those again, but that's the eosinophil. The basophil is an interesting little character. It's actually found in the lowest concentration in blood of all the leukocytes. And it acts similar to the mast cell, except it's found in blood as opposed to extracellular space or tissue. And it binds IgE, releases the granules that contain vasoactive mediators, but again, it's in such low concentration that its importance is really uncertain. That's the basophil. It used to be thought that the basophil and the mast cell were related, but it turned out that's not true. Although they have similar effects and they do similar things, they actually aren't related. They don't come from the same precursor cell. The mast cell, it resides in tissue than the extracellular fluid space, okay? And it binds IgE and degranulates to release histamine, and it's very important in the inflammatory response. There's a major role there, and so that's what we'll talk about him, the mast cell. The B cell, okay? The B cell is an adaptive immune cell. It's a lymphocyte, and it expresses antibodies as receptors. It's called the B cell receptor, but it's essentially an antibody that just has an extra transmembrane portion attached to it. And what that functions to do is recognize free antigen, recognize protein or chemicals or any type of antigen, anything that's foreign, a B cell would recognize. Of course, that B cell interaction is specific. Any one B cell will only recognize one antigen, okay? And then once it recognizes the antigen, it can differentiate into what's called a plasma cell, an effector cell. That plasma cell is differentiates from the B cell receptor and secretes all antibodies of any isotype. We haven't talked about isotypes. Isotypes are just different types of antibodies. But it'll release all the antibodies possible. Of course, those antibodies will be specific then to that one antigen that was originally recognized. Then you have, okay, that's the plasma cell. You got the T cell, and there's two major subtypes. Major. There's more than just two subtypes, but two major subtypes are the cytotoxic T cell and the helper T cell. Cytotoxic T cell is interested in killing damaged, infected, or cancerous cells. And the helper T cell is involved in then activating to give a very robust adaptive immune response. It's very important for activating an adaptive immune response. The helper T cell, if you don't have helper T cells, you'll be dead very shortly. Okay, it is not just like some mommy little helper. The helper T cells are very important in exacting a very strong adaptive immune response. One last T cell to mention, I guess a minor subtype, is going to be the suppressor T cell. The suppressor T cell is involved in what's called positive and negative selection of the T cells, and this happens during development up until puberty. The T cells are found within the thymus. The suppressor T cells found within the thymus are involved in then making sure that the T cells will be able to recognize healthy cell and bind to it, but not bind too tightly so as to kill it, and have the proper receptors to then bind antigen. If they don't have any of those things, then the suppressor T cell is going to kill either cytotoxic T cell or helper T cell. So that's positive and negative selection that occurs within the thymus, and the suppressor T cell is involved in that. Now, I'll never go through that, I don't think, at least, but just keep it in mind. And then we have the NK cell, which again is a lymphocyte, although it works in innate immunity for the most part. And the NK cell is in a similar business to that of the cytotoxic T cell. Okay, it's about killing infected, cancerous, or damaged cells, and it actually uses the same killing mechanism as the cytotoxic T cell, which is using perforin and granzymes. And that is the immune cells again in a nutshell, and we'll go through them later on in some detail for some of them, and other than we won't talk about them at all. One thing that I want to mention now, there are things called antigen presenting cells, and you won't be able to appreciate what this means until the adaptive immunity lecture, but I'll say it now. It's kind of a misnomer. It's a misnomer because, you know, when the good folks, the good researchers did immunology years ago, they were studying the CD4 plus helper T cell, and they said that antigen presenting cells were cells that presented to CD4 plus helper T cells. And those antigen presenting cells happen to be the dendritic cell, the macrophage, and the B cell. And so that's what they call the antigen presenting cells. But it turns out that there are other T cells. All right, there are cytotoxic T cells, for example. And those cytotoxic T cells will recognize antigen presenting cells as well. And it turns out that an antigen presenting cell for a cytotoxic T cell, every nucleated cell will present to cytotoxic T cells. So it's a misnomer. But traditionally, the antigen presenting cell, the APCs, are the dendritic cells, macrophage, the B cell. And we can move on now. So now, by way of review, I'll go through it in a different format. So we have blood cell development via hematopoietic stem cells to give rise to red and white blood cells. So the myeloid progenitor cells, as I told you earlier, give rise to then all of the leukocytes except for the lymphocytes. And the lymphoid progenitor cells give rise to all of the lymphocytes. That includes the NK cells. The myeloid cells consist of red blood cells, which are hemoglobin expressing, oxygen carrying, and make up the largest component of blood cells. And it's not an immune cell. We talked about hematocrit when we talked about in the cardiovascular physiology lecture that hematocrit is the percent of volume that the red blood cells in your blood take up once it's been centrifuged. That's typically around 45%. 45% of your blood volume is taken up by red blood cells. So it's a big percentage. You got platelets. They're not cells, but they're fragmented pieces of megakaryocytes that aggregate at sites of injury to stimulate and promote clotting. And again, it's not an immune cell. Granulocytes are so called because of their appearance and are easily stainable organelle granules within the cytoplasm of these cells. And these are phagocytes with specialized roles. Okay. You got the neutrophils, which are the first major type of granulocytic phagocytes, and they can find, ingest, and kill invading fungi and bacteria. The eosinophils are a minor class of granulocytic phagocytes with specialized organelles that contain toxic products that can be secreted to help kill invading parasites. The basophils are similar to mast cells in the connective tissue, but of bone marrow origin and circulate in the blood. A minor class of granulocytes that is a poor phagocyte, but use IgE as a receptor and are full of granules that contain vasoactive signaling molecules that assist in immune response. You'll notice that the granulocytes have a mnemonic. It's NEB. It stands for me, it stands for New England Biolabs, but NEB is neutrophil, eosinophil, basophil. That's my mnemonic to remember that these are the granulocytes. NEB, N-E-B. Alright, part of the myeloid cell that come from the myeloid progenitor cell is the monocytes. They circulate in blood and develop into macrophages upon entry into tissue or the extracellular fluid space, ECF space. The macrophages engulf cellular debris and form material left over from infection. That's what they do. Once they get into tissue, they differentiate into macrophage. You got the dendritic cells, or the DCs as we call them. They are of myeloid origin, and they're phagocytic cells in the peripheral tissue that is constantly engulfing material on the lookout for pathogens like bacteria or virus-infected cells. When DCs find these, they travel to the lymph nodes to activate CD4 plus helper T cells to induce B cells to produce antibody to the antigen that was originally recognized. Again, dendritic cells are antigen presenting cells that are keenly involved in activating naive CD4 plus helper T cells. These are the ones that will typically activate them, and most important in activating the helper T cells. Yeah, okay, the other ones do too. So the macrophage and the B cell also activate CD4 plus helper T cell, but when that first initial activation, what's most important is that you have dendritic cells to do that. And now we'll get on and talk about the lymphoid cells. So the lymphoid cells, again, are the cells that are derived from the lymphoid progenitor cells. One of those cells are the NK cells, or the natural killer cells. So they're a type of cytotoxic cell. A cytotoxic cell is a cell that can kill other cells, okay? And what distinguishes them from the cytotoxic T cells or the killer T cells or the adaptive immune cells is that they don't require MHC for recognition and activation of cytotoxic activity. MHC, the protein we haven't talked about yet, but MHC, the protein that is required for T cells to recognize antigen. Antigen has to be complex to MHC so that the T cell can recognize it. T cells do not recognize free antigen. They only recognize the antigen that is found on MHC, complex to MHC complexes, MHC proteins, major histocompatibility complex. And so what's special about the NK cell that works in innate immunity for the most part, and it does not need an MHC protein to kill a virus-infected cancers or damaged cells. And the significance of that is that many virus-infected cells will actually downregulate the level of MHC, which is typically used to then for cytotoxic T cells to recognize and kill virus cells. But when they downregulate that, they can avoid the cytotoxic T cell, but the natural killer cell can still recognize them and kill them. And they use perforin and granzymes to do that. Perforin forms the pore in the membrane, and that induces apoptosis. You got the B cell. It's also of lymphoid origin. It's an adaptive immune cell. It's an immunoglobulin or antibody producing cell. Antibody, immunoglobulin, same thing. Of the adaptive, and those immunoglobulins can recognize free antigen. So that's good. And then that can kill, that can not directly kill, but can neutralize pathogen in a number of ways. You have the T cells, and those T cells express Ig-like T cell receptors. They recognize specific molecules. Again, there's two major subclasses. The CD4 plus helper T cell. It's activated by recognizing a loaded MHC2 complex on antigen-presenting cells. Those antigen-presenting cells being the macrophage, the B cell, and the dendritic cell. And once activated, it will stimulate B cells that have produced an antibody that will recognize that antigen. Blue42. That's a mnemonic, Blue42. You want to understand it now, so I'm not going to go through it. I'll go through it another time. Next, adaptive immunity. And then CD8 plus cytotoxic T cells are activated by recognizing a loaded MHC1 protein, major histocompatibility complex 1, on host cells, or MHC1 activated killing of host cells infected with intracellular pathogen, i.e. virus, that are invisible to phagocytes. These CD8 plus cytotoxic T cells kill infected cells. Okay, mnemonic red 81. I won't go through it now because you want to understand it. And that is lymphoid cells. That is cells in general now. Blood cells. So the innate immune system has three essential functions. The first is that the innate immunity is the initial response to microbes that prevents, controls, or eliminates infection of the host by many pathogens. Non-specifically now, it's very important that you understand that the innate immune system is a non-specific system. It does not care about pathogen A from pathogen B. All it cares about is getting rid of pathogen. And it will recognize classes of pathogens, and it will differentiate between classes of pathogens, but it won't differentiate between Streptococcus pyogenes and Streptococcus pneumoniae, for example. Okay, it doesn't care about which one it is. It just cares that if it's a coccus, perhaps, or if it's a gram-negative bacteria or gram-positive bacteria, but it won't differentiate further than that. The innate immune mechanisms eliminate damaged cells and initiate the process of tissue repair. That's also something that's much less appreciated by younger students, is that you can initiate the innate immune response from simply damage to tissue. You don't have to have any infection to initiate the immune response to get innate immune response to get inflammation. Okay, and then the third essential function is that the innate immune system stimulates adaptive immune responses and can influence the nature of the adaptive responses to make them optimally effective against different types of microbes. We won't be able to appreciate this in our lectures simply due to the fact that it would require too much of an understanding of different cytokines and their relationship to understand this, so we won't be able to get there, but we will come across both of these two very much readily and shortly. So I think it would be instructive to highlight the important differences between the innate and the adaptive immune system before we get into the innate immune system. Okay, so let's compare the two. So on the y-axis here, we have self versus non-self discrimination. We have the lag phase, we have specificity, diversity, and memory. And then on the y-axis here, we have innate, and then we have adaptive, and whether or not it's present or absent. Okay, so why don't we look at self versus non-self discrimination? For the innate immune system, of course, it's present. The reaction is against foreign substances, foreign pathogen, foreign antigen only. Okay, whereas the adaptive immune system is also the same. Okay, it only reacts against foreign or damaged. The lag phase, okay, there is no lag phase for the innate immune system. The innate immune system is immediate. It is right when a pathogen enters the extracellular fluid space or the body that you will enact the innate immune system, where the adaptive immune system takes a few days to get going. Okay, So the response, there is a lag phase with the adaptive immune system. Specificity, it's very limited. The innate immune system, again, is very nonspecific and will mount a response for a wide variety of agents. Okay, whereas the adaptive immune system, there is a lot of specificity there. And in fact, it's very, very specific. You'll only get an immune response if an antibody or T cell receptor recognize a very specific antigen. Diversity, the innate immune system, the diversity of the receptors found on innate immune cells is limited. Hence, you have limited specificity. Where the adaptive immune cells, you have a very large, extensive, diversified receptors found on the adaptive immune cells, and that confers its specificity. How about memory? There is no memory associated with the innate immune system. Okay, later exposures will generate the exact same response, where the adaptive immune system, later exposure, will induce an amplified response. That is the basis of immunization. So immunizations play on the fact that the adaptive immune system will enact a stronger response when introduced with the same pathogen later on in life. Okay, so that's kind of comparison of the innate and adaptive immune system. All right, so now we'll get into innate immunity. Here's the general overview, general picture about the things that we're going to go through in terms of innate immunity. So there's external defenses and internal. External, we're thinking skin, we're thinking mucous membrane, and we're thinking antimicrobial proteins. Okay, so some examples, well, skin, you don't need examples for skin or mucous membrane, I'm assuming, you know. But antimicrobial proteins, we're going to be talking about a few, such as interferons, defensins, and cathelicidins, complement proteins, for example. Internal defenses, phagocytic cells, antimicrobial proteins, natural killer cells, inflammation, and fever. So phagocytic cells, the prophagocytes, again, are the neutrophils, the monocytes, the macrophages, and the dendritic cells. It's not that the other immune cells don't act as phagocytes, but the four that I just mentioned, are the cells that primarily will act as phagocytes within the immune system. Okay. You also got the antimicrobial proteins. Again, they're found both ubiquitously, both externally and internally. You got the natural killer cell. Those guys, again, use a set of proteins called perforin and granzymes to then induce apoptosis in either virally infected cells or cancerous cells or damaged cells. And you got inflammation. We'll talk about that. Inflammation is an interesting little series of events that hinge upon a lot of physiology that is kind of basic physiological knowledge. And then we have fever. Fever is induced by tumor necrosis factor binding over there in the hypothalamus, causing then fever, increasing the temperature set point at our hypothalamus. So the question is, and something that I guess sometimes doesn't even dawn on people, is how do the cells recognize pathogens, their particles, or damaged cells? And how do they do it non-specifically when we're talking about the innate immune system? Because clearly, you know, we're saying everything's non-specific for the innate immune system, so how are we recognizing pathogen? We'll get there. It's receptors, of course, and nothing special about that. The receptors that innate immune cells have are called pattern recognition receptors, PRRs, pattern recognition receptors. They're primarily expressed by phagocytes, dendritic cells, epithelial cells, and those cells non-specifically now recognize pathogens